Hi, welcome, and thank you for joining our first particle panel webinar series where we design the next hour to be interactive and educational. So that is, we hope that you will learn something practical at the end of this webinar. Today's webinar is titled Inline Particle Size Analysis, Interpretation of Results and Correlation with Orthogonal Methods. There'll be more to come, so make sure you join our newsletter by answering yes to the post-webinar survey or simply connect with us on LinkedIn, Particle Characterization Group. My name is Julie Chen Nguyen. I will be facilitating today along with your presenter, Darren McHugh. Darren is the Icon 2 product manager at Innopharma Technology. He received his degree in mechanical engineering, and he is joining us today from Ireland. As a product manager, he's responsible for setting the vision and strategy of products. He's the voice of the customer and facilitates a talented group of players to help steer the product roadmap. We're very excited to have him speak and do a demo for us today. All right, it's all yours. Please go ahead and share your screen.
Okay, well, hello everyone and welcome, and thank you, Julie and the team at HORIBA for organizing this webinar. It's great getting an opportunity to present again. Today we're going to cover particle size analysis. We're going to be looking at essentially our Icon 2 technology and some orthogonal methods of how you can compare that data. Specifically today, we're going to be looking at Sieve, the traditional method. Without further ado, let's jump right in. Okay, so if we look at the list here of what we're going to cover, the goals by the end of today, ultimately, by the end of this webinar, we'll hope you'll have an understanding of the methods that you can use to validate your analytical data for your technology, specifically focused in this webinar on particle size with an emphasis on solid bulk powders in the micron range. But the method that we're going to be doing for correlation and comparison to orthogonal analysis, it's going to be universal, so in its approach. So there's something here hopefully for everybody. You can use other types of methods by just using the same types of steps we're going to use for correlation. And maybe hopefully you'll get something out of this in terms of next steps to establish what is required to go from maybe an analog method to a digital method, to progress from maybe to a more smarter, more efficient methods of analysis using newer technology. And essentially that's our mantra here at Innopharma Technology. We exist to bring manufacturing forward. So particle size, it's an important component in the efforts to formulate and manufacture pharmaceutical dosage forms, as I'm sure you're aware. We'll begin at a very high level, looking at what particle size is and why it's important, but we won't stay there very long. We're going to be diving deep into analysis, looking at how to compare this data, because there is plenty of literature already out there and videos available on what particle size is, and we have those available as well. So we hope you'll get something from this. After we have a look at that, we're going to do a bit of a live demo, quick, I guess, whip around here at our lab, showing some of the analytical technology for particle size. And then after that, we're going to go into our correlation. So we're going to actually take some of the data from our particle size technologies here. And I've titled this as Death by Excel because it will be a little bit of an analysis where we're pulling data together, looking at what it charts like, looking at what it comes out like, talking about the results in terms of its correlation, its regression, etc. And there'll also be a mention as well as how this all aligns with the FDA guidelines. Okay, so let's get into it. Okay, so it's a small matter of particle size. Well, particles can be considered the VIP in our industry, or as we would call them, the CQAs, the critical quality attributes. Particles make up everything on our daily lives. They're all around us, you know, in the soil, the food, the air quality, all these things are actually made up of particles. And these images that I've just shown on the screen are actually one from the Hubble telescope, showing the Abell nebulous or galaxy. And then the one on the right is just taken from our Icon technology, showing something at the micron range. So it's just to try and emphasize that everything around us is affected by particles and that they are critical. Okay, so knowing the particle size is to know how they influence the environment around you, but more on that in a moment. So why is particle size important and why are they king? Well, particles, first and foremost, affect our end user and also the end product. Now, for us in pharmaceuticals, that's the patient. Knowing and being able to control the particle size is delivering on efficacy. So the ability for the product to perform its task. So essentially we are a cog in that wheel of improving people's lives and improving the quality of life for people. And that should be a driving factor for what we're trying to do every day, I think. Then second, actually to, let's say, the end product for quality, another aspect of that of quality is the manufacturability of this product. So things like particle size affects things like flowability, blend uniformity, compactability. The link between particle size and product performance, again, is well documented. It's not what we're going to cover here today, but there is plenty of studies out there on how particle size affects things like dissolution, a study we've done ourselves, absorption rates, content uniformity, etc. So by monitoring and controlling particle size, you're ensuring consistency. And that's the goal in manufacturing. You want a consistent, high quality product so you can compete. Okay, so moving on from that. Little animation there showing that particle's king. Okay, so particle size methods. Unfortunately, there's not a one size fits all, and I'm sure there's a bit of irony in there as well. There is different ways to measure particles, and you will get different results. And that's, again, well documented, but the slide on the left here is just showing different ways that a particle can be interpreted. This is quite a good book by Dr. Sorry, my pronunciation is terrible, Henrik G. I recommend you read it. It's got a lot of good information about particle size. But this is just showing that if something was measured by a projection or its area, how it can be different. And then another way to measure particle size is what configuration you're measuring it in. So these have their advantages and disadvantages. So when selecting your particle size method, there's a lot to consider. Is it an inline method? Are you measuring right in the process where the particles are being made? Is it an online method where it's passing by a sensor maybe and going back to the process? Is it an at-line method or even an offline method where you're drawing samples down to measure critical quality attributes such as size? Then particle size itself, this is something we cover quite a lot, but what is it, the industry standard for measuring particle size? And that's D values, D being diameter values. So on the right, we can see a bunch of particles of different sizes and shape there in the green. But if we wanted to define what that was, you wouldn't just take the average measurement of all those particles. You would actually look for the particle size distribution, which will give you an idea of the length and breadth of the particles that you're looking at. And in the industry, for a quick analysis, that's always been the D10, the D50, and the D90. And what I mean by that is, if you look at something like the D10, when you line up all these particles in a row, you're looking at the 10% weight or the size of these particles. And then the D90 is where 90% of the particles fall on that size. And then in between that, of course, is the D50, which is essentially your average. Very important to be looking at these for your process, because you can tell things like if you're getting an increase in the D90, maybe that you're generating agglomerates with your process, or at least your particles are rapidly increasing until your D50 levels out. So they can be early indicators for process parameter changes. So that's particle size and how it's measured. But then there's different ways of measuring those particles. There's a few industry standards shown here. You can do a projection, or you can actually take a chord length from that particle. So on the left here, we're looking at XE min, which is essentially the shortest chord length of an irregular shaped particle. And this is something that aligns very well with sieve analysis. And I'll explain why in a slide or two, why XE min, the shortest chord length, is something that's very close to what you get as a result of sieve. But what you do with that XE min is you actually convert it into equivalent sphere, and that's how you work out your volume. So the systems that use XE min will work out a equivalent spherical volume of that chord and work that out. There is other methods. You can take FE max, which is your ferrite chord length, horizontal or vertical, the longest length that matches it. And then on the right is actually what we use here for our icon technology. The image analysis we use actually matches ellipsoids to the irregular shaped particles, where you're actually getting a major and minor axes, so a tighter fit essentially. As mentioned, it's comparable to sieve for XE min. And then shape parameters are something that can also be taken from image analysis. And I noticed that on the poll earlier, it was particle size and shape that were most interested to people. So that's good, because image analysis systems are very adept at also detecting shape analysis, unlike some other laser diffraction systems that can't do that. Okay, So if we look at the industry standard, sieve analysis, the oldest particle size measuring method in the book. Sieve analysis has been around since forever, and it will be around forever. It's the fallback on every company for measuring particles. But saying that, it's not without its own faults. So essentially how sieve analysis will work is you have a stack of sieves. You can only stack that so high, maybe about 8 or 10 sieves high. And all those sieves will have different meshes, these blue lines that you're seeing on the right, showing different apertures that particles can fall through. And what's left behind essentially is that particle size and above on that sieve. So I call it binning. You're actually binning different particle sizes onto those sieves. But that has its drawbacks for a few reasons. One could be your sieves could be damaged, but you can also get them certified. The other main one is, as you can see from this image, when the particles are shaken on the sieve, what happens is the smallest diameter or the smallest chord length really on that particle is what will fall through that aperture. So if you have a very elongated particle, which we're showing here, it's going to fall through that sieve and give you maybe a bit of a misrepresentative result on what the actual volume is of that particle. It's just the smallest one that fell through. But again, it's the standard method. Again, it's an offline method, so you have to take samples to it. It's somewhat analog in terms of there's human input required to put the results into some sort of database. So it has all those, I guess, slower drawbacks compared to more inline technology, which we'll be showing in a moment. Okay. So inline technology that we'll be showing in a moment, the Icon 2. So the Icon 2 is designed to tackle these issues that you see with things like sieve analysis. It's designed to be an inline instrument, where I showed the instruments earlier, or the configurations earlier of in and at line. The Icon is designed to be fitted onto your process, so you're getting real-time data. So you can make more informed decisions about your particle size. You don't have to send it off to a lab for analysis, maybe even post-process and then find out the results a day later or a couple of hours later. The Icon gives you the affordability to find out what's happening to your process in real time. It's a non-product contact device, and we'll be showing that a little later in our demo as well. But this is just to give you the specifications of what it's used for and the environments that the Icon 2 is designed to be used in. Top left, you can see it mounted to a fluid bed. Bottom left, you'll see it in its at-line configuration. So it also has the versatility of being an inline and converted to an at-line tool for an analytical lab if need be. It ticks all the boxes in terms of the pharmaceutical industry for GMP, 21 CFR, ATEX rating, IP rating, and it also can communicate over industrial protocols such as OPC. So that gives you affordability for things like automation or integration into other subsystems within your factory. A little bit more then about the Icon. If we compare it to, say, Sieve, it can be an in and out line method as mentioned. It's dynamic image analysis, non-product contact, real time. It's digital. It doesn't suffer from drift because it's a digital camera method. And there's no element of model building with a system like this. It really is just setting up a configuration so you can focus on your material, have it correctly installed, and you're able to measure particles continuously. Okay, so it removes or reduces the need for sampling. So that can be a huge benefit for something like maybe sieve analysis, where you're very heavily dependent on those results. You could get this data in real time, so you can do more things like just-in-time release or basically improve efficiency in your process and get things to market quicker. Also, you get insight into what your critical process parameter changes are doing to your critical quality attribute of particle size and shape. So if we had this mounted onto, let's say, a fluid bed or a twin screw, and you adjust something like liquid addition rate, you will see what effect that has on your particle size in real time, something you can't do with other at-line or offline methods. It's also, we say it's Pharma 4.0 ready, and that really comes down to things like the OPC or being able to be connected via WebSockets. It is a smart device that can be plugged into a Pharma 4.0 architecture, so it can communicate over your industrial protocols for IIoT, etc. This is just a little animation showing how it works. So we have particles that come by the front of the Icon, and in there we essentially have a digital camera and our illumination ring. It illuminates the particles, there's a flash image, and this little circle with the particles you see in the middle bottom is a raw image that you get back from our system. So unique aspects of the Icon is that it has a crossover illumination method, and it can accurately do edge detection of particles that are overlapping. So if we talk a little bit more then just about the method of operation of something like the image analysis versus maybe Sieve, we mentioned before a little bit about chord length or XC-min. The top left here is showing traditional methods of determining a size for particles. You can take something like ferret's diameter, the longest horizontal chord length, or you can take something like Martin's diameter, the shortest chord length horizontally, and project them as a sphere, and that's an equivalent spherical diameter of how you work out particle size. There's plenty of systems that can do that. If we look at the images in the middle, the top is just a raw image of 3 particles from the icon. If we look at the middle image, it's with the blue spheres, that's an equivalent spherical diameter being matched to them. But if you use the elliptical method that our system does, you get a tighter fit for irregular shapes, and you get a major and minor axis from that. So from that major and minor axes, you're able to take the average of them for your third dimension, and that's how we work out volume. And our system does that in real time for every image. All right, moving a little bit on then, just from, let's say, the types of particle size analysis that are out there, the orthogonal methods, and a little bit on from the ways you can actually measure particles. If we actually talk a little bit about guidance documents from the FDA, so we have the analytical procedures and method validation. This might be familiar to some of you, but it's a great document if you haven't seen it. It basically shows a path of how you can take maybe an existing method compared to an orthogonal method to build a case really for bringing that method into your factory. So why would you do something like this? Why would you do this analysis? Well, maybe over the life cycle of a product, new information and assessments might become available. You know, like Sieve's been around for centuries. New digital methods are coming into vogue. They're, you know, inline to real time, for example. So you might get better understanding of your CQAs and what's happening in your process by bringing them in. But and this may warrant then the development and the validation of a new alternative analytical method. And that's what this document really highlights. So there's just a statement here talking about really new technologies that's mentioned in this document. But essentially under alternative analytical procedures, the data that you generate could be used as part of an analytical method to a comparability study. So you can bring more technology to the forefront. And also, there's a lot of companies out there who are integrating newer technologies to stay more competitive. They want to leverage these new studies to stay on top of their game. So they might be finding out things like how they can improve yield or how they can reduce batch time, etc., from measuring in real time. Okay, so that was a real quick run through of essentially, you know, what are particles, how to measure particles, different methods of measuring particles, and why you might switch from one particle method to another, and how you might actually go about doing that with guidance from the FDA. Next, we're just going to switch over to a little bit of a lab tour here, showing some of this hardware. So I know Julie mentioned at the beginning, but welcome to our process lab here in Dublin, Ireland. This is where we test all our analytical equipment. We also do a lot of solid oral dose processing here as well. If I take you over here, we can actually see the aforementioned sieve analysis. I'm sure a lot of people are familiar with it, but it's basically just to show in real life what it is. Essentially, sieve analysis is a stack of sieves, varying grades of different sizes. So you put your material in the top, maybe 100-gram sample. This will shake for a set time, depending on the SOP that you set out. And then you weigh all the sieves before and after to find out how much weight is sitting on each one of these stacks. From there, you'll be able to build a cumulative distribution curve, and we'll show that a little later on. All I really want to show here is that these sieves are just essentially made up of wire meshes. You can get them certified and regularly calibrated, but sieve analysis is a very robust method, very long-standing, but it's not without its drawbacks, as mentioned, mainly to do with orientation of particles potentially falling through on their shortest size, not on their correct size. Okay, so if I move this guy out of the way. All right, so behind me, you can actually see a bit of process equipment. This is our GPCG2. It's a fluid bed granulator, coater, and dryer. Today we have it running in its coater configuration. And if you haven't spotted it yet, this little green cord is leading up to the icon 2. and it's measuring real time in this process. So if we just take a bit of a closer look at what's happening inside that bowl. Okay, so from this angle, we can see the icon 2 is mounted inline. If I just point out a few little bits, this is the business end of the icon. In here, the digital camera is pointing into the process. It's mounted just via this flange here, which connects onto a pre-existing process window, very similar to one up top. And we can see that it's actually non-product contact. There's a gap here, and you might be able to see the flash of the LEDs as well, just taking images every couple of seconds of the particles inside the process. You can see from the long operator window that the material's fluidized, so it's constantly passing by the window. So we're getting data every couple of seconds on what's happening here. So if we change something like airflow, liquid addition rate, air temperature, we could measure that in real time, what's happening. At the back here, we can see the green communication cable, and that will run over to your process area where you'll have maybe an industrial PC or something like a laptop to read that. But if we actually have a look at what's happening with the software from ICON, which is called iPass, and this is actually what's measuring in the process right now. So this inset is just showing the real-time trends of your particle size. You can see these colored lines, the green, orange, and blue. That's your D10, D50, D90. So from this, initially, we can tell straight away we had a little bit of growth at the beginning. It seems to have leveled out now. If we wanted some more growth, we might have to do something like up our liquid addition rate. And this is the real benefit from using tech like this. This is something where traditionally you would either have to take a couple of samples, pass it by a sieve, assess whether your particles are growing. So it's giving you much more automated real-time data. Okay, so that's iPass running there. So if we just actually head back over here as well, just to finish up before we go into our analysis. One other thing is just to show that that was the icon in its outline, sorry, my apologies, inline setting, and this is the icon in its outline setting. So I mentioned before it was designed to be inline, but we have retrofitted it as well to be an outline tool with this automated sample presentation stage. So you can use this in an analytical lab as well. Maybe you have downtime on your equipment or it's been serviced, etc. You can fit it with this automated sample presentation stage and take static images much the same way you would with a digital microscope. And that reads out to our software here again, iPass. Again, the green cable just runs over to this communications hub here. That's just taking the Ethernet and serial communication, converting it so it can go back to the laptop here, which is doing all the processing. This is doing the real-time analysis right here. Okay. So we're going to jump back in now actually and do some analysis. I'm going to take some data from our icon, from the inline icon actually, and we're going to take some data from Sieve analysis. We're going to pull them into an Excel sheet and show what kind of correlations we can do. So hence this section was called Def by Excel, but I hope you stay with me for this and you get something from it. Okay, so let me just go back to sharing my screen. Okay, so here I essentially just have an Excel spreadsheet that I'm going to walk you through. What we're going to do today is this template is laid out for 4 different samples, but we're going to be looking at CSE 117. It's going to be pre-filled for us, so I'll walk through what that data looks like for an icon inline, as well as Sieve analysis data and how you compare the two of them. And then we're actually going to fill this one out ourselves, CSE 118, and we'll do that together live, so you might get a bit more of appreciation for how you do a correlation on that. On this first page, we just have some basic useful information on what correlation analysis is. But if I jump down to the chart here, it's a bit easier to explain in a simple example first. So let's say we have samples A, B, and C. We have 5 different measurements for those samples. And if we look at A, and that's the blue line here, we can see that it's got some peaks and troughs measuring across it. And then if we look at B, it doesn't match so tightly. And what we can do to that once we have all the data populated is we can do a correlation analysis in Excel. I suppose this is a good point to mention, this doesn't have to be done in Excel. It's whatever package you're familiar with. Maybe you have a preference in your company, such as MATLAB or R, or along those lines. But it's just I'm familiar with Excel, and it allows me to break down the analysis a lot more to the fundamental levels like this. So the first formula is just Corel, if we can see that up here. If we click on that, it's just showing we're pulling a matrix really of the A and B together. And when we look at this table, what we can say is obviously A compared to A is going to be 1, but when A is compared to B, we get a 0.18 correlation, and that's considered very poor. And that actually makes sense when you look at the graph. So if you look at A, which is the blue line, and when you look at B, which is this orange line, you can see that they're not really matching over these 5 points. So that makes sense. It kind of validates what we're doing here, as in like, yeah, there's not much of a relationship between these two. And we would expect it to be between 0 and 1 for good correlation. But if we then look at A compared to C, and if I click on that formula, we can see it's matching much tighter. We got the gray line and the blue line that are following each other very closely. And we're seeing a 0.96 correlation. So that's very good. It's very close to 1. So that's giving us confidence that there's a relationship between those two variables. And then equally, if you look at B versus C, we also see a very poor correlation again. And that makes sense. The gray line is not very close to this orange line. Okay, so that's it on a simple level correlation, just with some random data. But that's what we're going to be doing with our Sieve and icon data. And then we just have some information below on correlation, regression, what we'll get into. But if we stick with looking at CSE 117, if I jump into the Sieve analysis, and I said this one's going to be pre-filled for us, so I'm just going to walk you through quickly what this is going to look like. Essentially, just to describe these tables, I don't want you to get too bogged down in them, but anything in red is input data, and everything else is automatically calculated. I purposely left this Excel workbook very manual. It is something that you could probably, macro or automate if you had a very repeatable process in your company. But I really wanted to show where the numbers were coming from and where they were going today. So we have our Sieve analysis. This is very standard for what you would see. A stack of sieves of varying sizes, and then the material poured in the top. It was about 100 and let's say 4 grams, 5 grams, and this was the amount of material left behind on each sieve. From there, you can basically do an auto calculation then of what percentage of material was retained, the cumulative percentage retained, and the cumulative passing, which is what we're mostly interested in. So if I actually go over to the chart to explain it a bit more, so this S curve is showing the amount of material with these triangles left behind in each sieve. If I click on that, you can see where the data is coming from. To determine the D50, the D90, and the D10, what you look for is where they sit on that curve. So let's just look at the D90. It's coming out as 594, but because of the bin size that it fell on, if we look back at this table, it fell, materials left behind basically on the bin between 900 and 500 microns. So that's why we have this error bar here. D90 is determined to be 594, but essentially all the material that was measured there is between the size of 500 and 900 due to the resolution of your stack of sieves. Okay, so that's a quick sieve analysis really for CS E117. What we do with that then is we actually interpolate it to get the full particle size distribution, because we need to be able to compare it to icon, which automatically measures the full size particle size distribution. So what we're doing is a straight line between the known points, and this formula here really will work out that data. So if I just click this one here, it shows the formula of where it's getting its values from. And if we actually click on the determination of D10, we can see it's worked out from these values here. And that's validated for us because I can see this interpolation is working out to match what was determined for D10, D50, D90 for what falls on the curve. So that validates that our interpolating of the full particle size distribution is pretty accurate for measuring against what the bin sizes of the sieve were. Okay, sieve analysis, that's auto-populated for us. That was a talk through in that. If we look at the icon data, so this is actually raw, let's say, icon data. It's a CSV file that gets output by the system. Each one of these lines is a image analysis. It's the particle size for that image analyzed at that time point. What we get here at the top, though, is an actual summary of all the particle size added together for the last 300 seconds, for the last 5 minutes. If we just jump here to the D5, this is our full particle size distribution right here, all the way up to 100. And what we do with that is we just clean it up a little. I'm going to take it to the next sheet. If I jump in here, that's these values in the red that we essentially took from our icon file, dropped it in here. We just do a little bit of data cleaning. We transpose it, we plot it just to see what it's looking like on our S curve, and we determine the D10, D50, D90. That's all just before I take it to our last step really for correlation. So we've got our sieve data, we've got our icon data for CSE 117. And then we actually correlate. So if I jump to the correlation for CSE 117, we have the full particle size distributions here for Sieve and Icon, and you can actually see they are measuring differently. So what's important here is what we covered earlier, saying that all particle size measurements are not created equally. Sieve measures differently than Icon. Icon measures differently than other methods on the market. So it's not really an apples for apples comparison. That's not what you're looking for here. And it's not what you should be looking for here. You should be looking for the correlation value. What is the relationship between the Sieve analysis for this material and what is the Icon relationship? So we're going to do correlation again, very similar to as I shown on the README. If we just click on this, we can see that we correlated the particle size for Sieve and Icon. It's coming out to be 0.9964, which is great. It's showing a really strong positive correlation. But what does that really mean to us as well? Okay, we can actually see there's a relationship between the two of them. But does it matter? Is it statistically relevant? What's happening to trend? So correlation is great as the first step. It'll be part of your story toward your analysis. If you see a positive value like this, if you see something maybe negative, that this value was a minus, or if you've seen something that was probably below a 0.5, you would see moderate relationship between the two values. It would be showing that there's issues, that these two sets of data have no relationship to each other. So that could be for several reasons, but we won't get into that now. What you do after you correlate is then you can look further into it by doing a regression analysis, essentially looking for a straight line between all the values and working out your R squared. And the regression analysis takes it a bit further because it can independently look at the relationship between, let's say, A, which is Sieve, and the relationship between B, which is Icon 2, something that correlation doesn't do. So this next step here, and this is automatically done by data analysis in Excel. I'll show that a little later on how you do that. Right now, it's already here for us, and I'll just talk about the values. So if we look at what's highlighted, we can see our R squared for this is 0.9928. What does that mean? Where did it come from? If I jump over to this chart a little bit on the right, we can see Icon plotted against Sieve, and these are all the X's on the chart. And then when you put a linear regression line, a trend line between those, you get an R value of 0.9928. So from that straight away, I can say that it's a positive relationship. It's a strong relationship between the two independent variables. Again, adding to this analysis that there is a relationship between Sieve and Icon. It's looking positive. If the question comes up then, but is this significant? What is the actual bearing of having these values, such as correlation and regression, being positive and strong? That's where the goodness of fit, like an ANOVA analysis, will come in. And you can look at mainly two things here. You can look at the significance of F. If that is below 0.05, it's indicating that the results you have are significant. There is a relationship between them. And then you look at the p-value as well. And the p-value is the probability relationship. So if this is also below 0.05, you're essentially saying that the probability that this relationship is accurate is 95%. Now, 95% sounds good, but you always have to have in the back of your mind that there is also a 5% chance that this is coincidental. And that's why you do repeat studies and expand your study. Okay, so that's me talking through a pre-populated analysis of, let's say, Sieve versus Icon. Next, we're going to move on to CSE 118. We have no data here. We don't have anything for our Sieve analysis yet on CSE 118. If I just move over that part of the table. So we see there's nothing there. So essentially, we need data for that. What you do, depending on what method you have, will open up some Sieve data. So Sieve data could typically look like something like this. Depends on what way you measure it. So we're looking for CS118. Here's the size of the bins, and here's the weight of the sieves after your sieve shaking. So we're just going to take that, go back to our sieve analysis. I'll drop them in there, and the rest is automatically calculated for us like before. So we have the percentage retained, cumulative, retained, and passing, and that changes our chart here on the right as well. We can see the cumulative curve with our D90, 50, and 10 values, where they sit. Again, with the caveat of the error bar here showing where they sit in that analysis. Then we have an automatic interpolation as well for the full particle size distribution, as mentioned. So that's our sieve analysis. That's going to be populated all the way down to the correlation for us. Next, we'll take the icon data. So we get that. Icon exports as CSV files. I'm going to do CS118. Alrighty. So we're looking for the these two lines here. Essentially with our full particle size distribution. I can drop that into our template here under cleaning of icon 2 data. When I go down to CS2118, I just want to drop that into essentially this line here, and I'll move down a bit so you can see it all automatically populate once it's ready. Bear with me. Okay, that's great. So this is just a bit of a data clean. We're basically plotting the full particle size distribution with the D10, D50, D90s. And then we can now jump back to our correlation tab. So all these numbers are being automatically pulled from those sheets I populated. We see sieve versus icon side by side. And then we can go straight into looking at the correlation between the two of them with the same formula for working that out. Sorry if I click that. So this time it's actually 0.8994 to be accurate. So again, a very strong correlation for these two materials, which is great. As mentioned, if this number was coming out, let's say maybe negative, or if you had a couple of, let's say, crazy outliers, for example, I will just simulate that. These values will drop down, maybe even if I go further. Let's say we had 1,000 in there. So it's getting less significant with so much variation. Okay. So that's showing that there's a strong relationship independently between icon and sieve. That's a great first step. So I think it makes sense now to move on to looking at the regression analysis. And I'll just show how you do that with Excel. I'll click this here. So this is actually a plugin that's probably not activated by default by Excel. You actually have to go to your, if I move this bar, you actually have to go and install this. So sorry, bear with me. Oh, it seems to have been even deactivated for me. Okay, this might be a good example to show how you actually activate it. So we can go to options here, or add-ins, analysis tool pack, make sure that's activated. And then you actually get this little bar added here under your data tab. Okay, so with that, I can select it. It gives you lots of different options for what type of data analysis you want to do. Today, we're interested in regression, so we'll select that. We'll select our input range for Y, we'll put that as the sieve. X is going to be the icon. And then I just want to show where I want it to output. We'll populate those cells here, for example. And if you're interested in looking at other things like residuals, you have those options to tick here, but for simplicity, we're just interested in the ANOVA analysis. And then that populates that there for you. So looking at this, the R squared is coming out to be 0.9788. Again, a very strong relationship showing that there's a good positive linear line running between these 2 points. And we can look at this chart here to verify that. So we have icon plotted against sieve, a trend line added in between them, and it's 0.9789. That's great. We can look at the significance of F. We actually have to convert that to a number. That's great, showing up as 0. So if it's below 0.05, it's showing that the significance of F is relevant. And then also, we'll just convert these to a number quickly. And they're also below 0.05. So that's great. It is showing that between a correlation and a regression analysis for our, let's say, sieve versus icon, or equally, if you wanted to add in a third parameter here for some maybe other analytical method, you could do that and expand your matrix here. So this is great. It's showing that icon and sieve for these particular data points are showing a strong relationship independent of each other as well, showing that if you wanted to expand your study, there's validity here to do it. Or if you wanted to use this as part of a filing, that you can do that as well. I guess one other point as well, the difference between correlation and regression is regression can also be used for prediction. So you can actually use forecasting with regression analysis, something you can't do with correlation. Okay, so I hope that wasn't too boring or too fast for everybody. It was just really to show where you get your data from, how you can drop it in here, how you can do some simple analysis with it in Excel, and this can become your justification or part of your filing if you were looking for an alternative analytical method, if you wanted to bring in something like inline technology, that's only where you only have maybe an at-line technology currently. So let me jump back to some slides just to finish up, and we'll take some Q&A after that. There we go, cool. All right, so we moved on from the lab tour. We've covered a little bit really about correlation and regression analysis. I did mention, this doesn't have to be done via Excel. It can be done via your statistical package of preference. You might have something already existing in your company. But just because there's a straight line on the graph, that doesn't necessarily follow that the one thing caused the other. There's the classic saying, correlation doesn't imply causation. So you can assess the level of confidence by calculating your regression value, your R2 as we've seen. And in these two cases, we've seen that it was positive, it was above a 0.9 value. If it was below maybe a 0.5 or even a negative value, you would see that there is no correlation between them. So you would either have to reevaluate your testing method or come to the conclusion that there is no relevance between these two pieces of data that you're trying to compare. So we were able to tell from just looking at our regression values whether the relationship between ICON and Sieve is statistically relevant. And for that, we ran a statistical correlation test. We seen that P was below 0.05. So that gave us a 95% confidence interval that this data was statistically significant. That's essentially it. This slide is just showing some of the app notes and stuff we have done to date, or our case studies. We have done comparative studies of our technology before versus others. We have done dissolution testing, seeing what effect particle size has on that. So you can look into those further. That's it essentially from me. I think we'll open it up to some Q&A.
Hey, thank you, Darren. Thank you for this really good talk and demonstration. I'm actually really impressed by your Excel skill. If you have other questions, please don't hesitate to reach out to us at labinfo@horiba.com for any questions or feedback that you may have. So let's hop into Q&A session. A question that came in is, any way of measuring the specific surface area of large granules with this equipment?
Yeah, good question. Unfortunately, short answer is no, as the current technology stands. It is designed for particle size and shape, but technically, it could be possible with further development. It's not something we're specifically, let's say, selling this instrument toward, but I'm just speaking because it uses a crossover of light. You've seen the red, green, and blue particles. From that, you would be able to map a topography of the particles, and using maybe some post-analysis, some other methods, you would be able to determine surface area. But I do say that as additional research if someone wanted to pursue using ICON 2 images to do that, they might be able to do so. But the technology as it stands is specifically designed for inline, real-time particle size and shape.
Okay, so how do you integrate the sensor with HMI, human-machine interface of process equipment?
Sure. Yeah, another good question. There's lots of options. We can, a lot of our customers might have their own HMI on their process equipment that they want to use to look at their particle size and control ICON per se. And we can do that either maybe very simply via things like OPC, Open Platform Communication, where we take the data from the ICON and display it on their HMI, or we could go further and implement like a WebSocket, which will allow you to take IPass essentially from the PC that it's running on and display it on your HMI if you wanted to do it that way as well. So we have various options. The unit comes as standard, let's say, with a laptop, but if it was to be installed inline, we would work with the automation engineers or the IT department to implement it onto their pre-existing HMIs.
Great, thank you. Now, how can you discuss a little bit on how is the method validated?
Yeah, good question. And the answer is depends. So we did talk a little bit about maybe validation with what we just discussed about how you compare, let's say, ICON to an orthogonal method like Sieve or whatever other at-line or inline particle size methods you might have. That would be one step toward validation. It is somewhat company-centric. They do have their own validation plans from the interpretation of FDA guidelines, and we can work with those as well. So we have seen different ways of doing that. We also do provide verification kits, size verification kits with the equipment, meaning basically if you put a known size standard underneath the instrument, you'll get a known result back. That's another method toward validation as well. And that's part of our installation or operation or qualification packages.
Thank you. Okay, so a major challenge in particle size is often touching particles or agglomeration. How does ICON address this concern?
Sure. Yep. It's a good question as well. So there's two ways to look at it. If the ICON is measuring in its benchtop setting, it's using a static image analysis. Now, for that setting, you have to present a monolayer, a single layer of material to the camera system. So there's no overlap of particles. With this inset here, you can see it. We just have our the benchtop that I showed you earlier. We're looking at some cellets here. They're not sitting on top of each other. This is purely because of the static image analysis, where the stage will move, stop, the camera takes a single image and analyzes it. It's not so critical for dynamic image analysis, which is what we use for our inline settings, which is what you're seeing here now happening with the GPCG2 on the coating process. If particles are in the background or sitting on top of each other, because of our illumination method, where you can see the red, green, and blue crossover of light hitting each of the particles, we're able to do edge detection of where maybe half a particle is sitting behind a particle and correctly match to the two of those. So it does depend somewhat on what method you're analyzing, but also things like we will consider for integration would be sample presentation and representation. And maybe this is a little bit outside of overlapping particles, but we would look into it in terms of what the material is being presented to the camera system. Is it representative of the process? Are we getting the full particle size distribution? And also, is it an accurate area to measure in terms of, there's no blockages in the process or anything like that?
Thank you. That's perfect. How does ICON 2 differ from other dynamic imaging systems, such as the one that you mentioned in your spreadsheet, the Camsizer?
Sure. Yeah, there is other methods out there. I guess some of the unique methods from the ICON system is, first, it uses elliptical fitting method. So that's where it takes the major and minor axes from an ellipse fitted to the particle. So that works really well for irregular shaped materials. And then also the illumination method of the red, green, and blue combined with the color camera also give us the advantage of overlapping particles for edge detection. So there is that versus some other inline methods that might use a backlit method, where you're seeing a black and white image, and where crossover of particles would be an issue there, because let's say a very large particle which would come up black on a white background, there might be another smaller particle inside that large one that you're not seeing because it's monocolor that you're seeing. So that is some of the advantages of the ICON system. It's elliptical, non-product contact, and it's also crossover of light.
Perfect. And I think the fact that you can measure particles in real time is a huge benefit as well. Okay, so the next question is, have you tested this technology, ICON 2, in a dynamic grinding or sifting environment like flour mill?
We have done various mill integrations. A flour mill is an interesting one because flour is a very difficult material to flow. And so what we use as an example for bad material quite a lot. But when it's being sifted, or maybe it's coming out of the mill where it's being processed through a sieve and it's being fluidized, that would be a good point to measure flour material with the ICON system. Once the material is within the size range, which we've seen in the spec sheet earlier is 50 to 5,500 microns, and the material was free flowing, we can measure it. So that goes back to the presentation and representation, how we integrate the ICON 2. The image we just insert now is just a couple of examples of material that you can measure. We're looking at powders and solid bulk materials. So they're all either free flowing, either white or off-white in nature, because of the illumination method, the red, green, and blue LEDs. We need to be able to illuminate the material. So We don't have a case study for flour. We have done a lot of milling integrations with pharmaceutical integrations, such as roller compactors, the exit of a mill there, which has all been excipients. But we would be up for a trial on a flour process once it was free flowing. I see no issue.
Thank you. In fact, may I add that for flour, because it's so fine, we often recommend a laser diffraction as a orthogonal techniques, just because it offers air pressure to disperse very agglomerated or very sticky material. Okay, so the next question is, how accurately does ICON can measure the particle size of moving particle? What is the maximum speed of particle where the readings are accurate?
Maximum speed. Okay, well, I guess there's two questions there.
Yep.
The accuracy of the particles, we have a spec sheet for that. With the verification standard kits that I mentioned, we supply sizes from 50 microns to 5,500 microns, and they all have their own tolerances because that's the way they're manufactured. We're able to measure within those specified tolerances. So, you know, maybe for 50 microns, we could be talking plus or minus 10 micron accuracy, and that gets a little bit larger as you go up. But if the person that asked that question is interested, I can send that sheet over with that full range of accuracy for all those sizes. To the second question about speed, I presume you're talking maybe about motion blur there, or what would constitute too fast a moving particle for the ICON system. We've used the system for up to 10 meters per second moving material, so that's not to say that's the fastest material we can measure. That's the fastest material we have measured, and it's still accurate. We have yet to find a faster material. So next we'll be talking about bullets after that. But no, it can accurately measure very fast moving particles with no motion blur. We're talking about a high shutter camera here, digital camera. So we've never had an issue with blur.
Thank you. Okay, and then for the next question, I can definitely answer yes. To evaluate for the instrument, we do accept sample analysis. Okay, I think that's all the questions for now. I will definitely relay these questions over to you, Darren, at the end of this webinar. And if you have any questions that you think of at the end, you can also email us at labinfo@horiba.com for any questions. Okay, so I think that's it for now. Is there anything else that you would like to add, Darren?
No, I just want to say thank you again. Thank you to everyone for listening. And as Julie said, if you have any questions, post webinar, please feel free to contact us. Happy to answer them. Yeah, have a good day, everyone.
Cool. Okay, we'll see you at our next webinar on April 29th, where Dr. Willie Hendrickson, the CEO and founder of Avika Group, will give you a very relevant talk on fluid bed processing, focusing on drying, agglomeration, and particle coating. I hope to see you guys there. Thank you so much. Bye, Darren.
All right, thanks. Thank you, Julie. Thank you, HORIBA.
Thank you.